Okay, PUC is the another plasmid, is the more advanced vector. Which structures allows the direct visual selection colonies containing the vectors with donor DNA insert. The key elements is a small part of the E. coli beta galactose gene. Okay. Uh, into this region has been the inserted the piece of DNA is called polylinker, multiple cloning site or multiple clone site, which contain the many unique restriction target sites useful for the inserting donor fragments. The polylinker is the frame translation only with the beta galactosides fragments and does not the interfere with its the translation. The transformation protocol is the receptor cells that contain the beta galactose gene lacking of the fragment present in the on the plasmid. And unusual type of the complementation takes place in which the partial proteins encoded by the two fragments unity to form the functional beta galactose. A colorless substrate for the beta galactose called XGOL is added to the medium and the functional enzyme convert to this substrate into the blue dye, which colors is the colony blue. If donor DNA is the inserted into the polylinker, the enzyme is fragment born on the vector distributed the no complement beta galactose proteins. Okay, the selection for the white AM colonies and the selected directly the vector being inserted in the such colonies are the isolated for the they can use for the further studies. Next one is the bacteriophase. Already we discussed the plasmids. Two types are the PUC, and next one another one is the PBR 322 is the plasmid. Another one is the bacteriophase. The viral vector in which the gene or genes is the insert in the incorporated into the genome of virus offer many advantages for the cloning and the subsequent application of cloned genes. The viruses infect cells with the high efficiently. The cloned gene can be the introduced into the cells at the significantly higher frequency than by the simple transformation. Viral vectors, such as the bacterial M13 based vectors, are the designed to the facilitate the sequence and the generation of the mutations in the cloned gene. The vectors derived from the retroviruses can affect the stable integration to the mammalian chromosomes of the cloned DNA, allowing the contain the expression of the gene. Viral vectors are also the vehicles of cloned DNA, allowing the continued expression of the gene. Viral vectors are also the vehicles of the choice for the gene therapy. Next one, phase lambda is one of the bacteriophase. Phase lambda is the convenient cloning vector for the several reasons. For lambda phase, heads will be the selectively packaged chromosomes about the 30 kilobase pair in length, and this the property can be used to select lambda molecules with the insert of the donor DNA. The central part of the phase genome is not the required for the replication of the packing of alpha DNA molecules, so that the central part of can be the cut off, but the using the restriction enzyme and the discard and B beta, the two arms of the are ligated the restriction enzyme, cut the DNA, cut donor DNA. Lambda phase the molecules 10 to 15 kilobases inside in the will be the most effectively packaged into the phase heads because of the size inside the substitute the central part of the phase genome. The second useful property of the phase vector is the recombinant molecules are the automatically packaged into the infective phase particles, which can be the conveniently stored and handled experimentally. And next one, the single standard phase. Some phase contain only single standard DNA molecules. On infection of the bacteria, the infecting strand is converted into the double strand replicate form, which can be isolated and the used for the cloning. The advantage of using this phase as cloning vectors is that the single strand DNA is the very substrate substrate required for the Sanger DNA sequencing technique, currently the widespread. Phase M13 is the one of the most widely used the this purpose. Cosmids. Cosmids are the vectors and the hybrids of the lambda phase and the plasmids, and their DNA can replicate the cell like the plasmid or be packed like the that of the phase. Cosmids can carry DNA inserts about 3 times as large as those carried the lambda itself. The key is that the most of the lambda phase structure has been the deleted, but the signal sequence that the promote phase had significantly. This modified structure enables the phase ends the stuffed with the almost all donor DNA. Cosmic DNA can be packed into the phase, particularly by using the in vitro system. A vector which is derived by the combining plasmid elements with the small M13 elements is called the phase mid. Phase mid is another example of the vectors. Is the P black script. The phase mid contain origins of the DNA replication for the both plasmids as well as the M13. As a result, they replicate as the plasmids E. coli, and they can also be packed as the recombinant M13 phase. Some most of the M13 phase functions are the not present, not present in the plasmid. The packing requires helper the phase to be present in the host cells. An example of the such as the helper phase is the M13 strain M13 strain K07, which contains all of the viral structural genes as well as the defective origin DNA replication. The origin of the DNA replication is the sufficiently active to permit the propagation of the K7 phase, but is the much bigger than the origin of the contained phase vectors. As a result of the infection of the phase made containing the bacterial cells with K07 helper. What are the application of the DNA technology? This is the ultimate goal of the recombinant DNA technology is to benefit the mankind of the livelihood. And some of the reasons the biotechnology is received to the much attention in recent years is because of the recombinant DNA technology. Before the advent of the recombinant DNA technology, there was only limited choice. Genetic manipulation is economically important organisms. Recombinant DNA technology has approved uh, manageable possibilities for the manipulation of organisms. Okay, what are the applications of the RDNA technology? Mainly the agriculture applications. Mainly agriculture applications is the mainly herbicide-resistant transgenic plants. Herbicide-resistant transgenic plants like the petunia resistant and the glycophosphate, tobacco tolerant, to the sulfonylurea, maize resistant to nitrogen have been the developed. Similarly, many crops, plants like the tomato, potato, oil, seed, rape, sugar, okay, beet, soybean, cotton varieties also be the developed. Herbicides, the resistant transgenic crops is already being the commercially grown in large areas of several countries. The next important is the insect resistant transgenic plants. Insect resistant transgenic plant crops is the carrying the Bt toxin genes have been the developed for the effective control of insect damage. The gene was the isolated from the bacterium Bacillus thuringiensis and inserted into the number of crop plants. Bt cotton variety resistance to bollard insect has already been the commercialized and the cultivated all over the world. Next one is the disease resistant transgenic crops plants. Transgenic crops plants the resistant to viral, bacterial, and the fungal disease are be using the developed transgenic plants. Have been the produced tobacco, tomato, and the potato is resistant the broad spectrum of the plant viruses. Coat protein CP gene from the TMV has been the transferred to tobacco and in the transgenic plant expression of the coat protection was observed in the papaya tobacco, papaya ring spot viruses that provide the coat protein gene. Potato leaf roll viruses, potato virus Y, potato and tobacco, potato viruses, rice strip viruses, tobacco, herbs mosaic viruses. And next one is the abiotic stress resistant transgenic crop plants. The serious crop losses are also the cause due to the variety of the biotic stress, like including the drought, cold, salinity, metal toxicity, etc. Transient plant resistant to the these stress have been the produced utilizing the transgenic that alter to the desired biosynthetic pathways. The next another important is the transgenic plants for the improved crop productivity. Okay, crop productivity improved. Production also be produced the model plant systems, demonstrating the possibility increasing plant productivity through the transgenic approach. Transgenic tobacco plants, which is the increased harvest index, have already been developed. And the crop plants like the rice, the use of the phytochrome gene for the increasing and harvest index may lead to the increased crop productivity. Transgenic plants for the nitrogen fixation effects are the being the made to the transfer nitrogen fixation and nip genes for the bacteria associated with the legumes to the other crop plants. The genes largely responsible for the process have been the cloned and the transformed to the gene plant cells, or the recipient cells have not been able to the fix nitrogen and successfully potential benefit for the crop plants such as the maize or the rice is a great. Transgenic plants for the improved nutritional quality. Transgenic plants have also been the produced that provide the functional food and the nutritional. The functional food is the define the food consumed as a part of the normal diet, but would we have the addition to the basic nutritional function, physiological fee benefit, risk chronic disease, and nutrient electricals is the hand of the product isolated from the purified from the foods that generally solid in the medicinal forms. Transgenic plants intended the functional food and the nutri- nutraceuticals, including those that would have be high concentration of the health promoting nutrients, including the improved quality of storage proteins, altered fatty acid composition, edible oil, variety of the vitamins, antioxidants, cholesterol, along the some of the plant produce the functional food is the nutraceuticals, substrate the pro vitamin A, the benefit antioxidant crop is the rice, Transgene is the phytoimmune synthase in the lycopene cyclase. Vitamin E antioxidants. Canola is the white tocopherol methanol transferase, the transgene. Substrate is the flavonoids. Benefit is the antioxidant. Crop tomato, colchicine isomerase. Fructose sulfates is the benefit is the low calorie. And the crop is sugar beet. On sucrose polyester. And the iron is the iron fortification rice. In the F10 phyto is the transgene. Extensive research in during the 96 in 2000 by Indigo Patricus is of Switzerland and Peter Bayer is the German is the developed the transgenic golden rice containing the beta carotene and the other carotene that are the precursor of the vitamin A. It is the believed to the one of the potential solution vitamin A deficiency in children. Similar efforts is being to the develop of the golden mustard. Okay, next one transient plants for food tasty and the appearance. The post harvest discoloration in the foods and the vegetables is the considerable problem for the food industry. Enzymes are the largely responsible for the discoloration and fruits and the vegetable. Recently, two commercial varieties, the potato and we developed the good level of the resistance in the enzymatic discoloration. Food industry, the foods are the appetizing factors like the sweetness, the desirable transient plants was produced much sweeter being developed. For example, transgenic plants, tomato and lettuce plants with the monolin gene encodes the fusion of the proteins that the produce the monolin is the protein this choreophyllum communis in the approximately 1 lakh times is sweeter than the sucrose. Transgenic monolin. Transient plants increase the shelf life. And number of transient plants, the different crops suitable for the food processing storage have been developed. For example, brucei-resistant tomatoes and the cantaloupes, melon fruits existing the delayed the ripening. tomatoes with the elevated sucrose and the reduced starch, tomatoes with the high levels of the total and the soluble solids, and potatoes with increased the starch content. Some of the medicinal applications of the recombinant DNA technology. Medicines have allowed the continue the major beneficiary of the recombinant DNA technology. The major areas which the medicine, the recombinant DNA technology can apply for the production of recombinant proteins, production of recombinant vaccines, diagnosis of disease, gene therapy and the cancer treatment, forensic science. First one is the production of the recombinant proteins. A number of human disorders can be treated the absence of the or malfunctions of the protein normally synthesized in the body. Most of these the disorders can be treated by supplying the patient with the correct version of the protein. But this the possible the relevant protein must be available in the relatively large amounts. Prior for the development of the recombinant technology, most human protein pharmaceuticals were available in only limited quantities. They were the costly to produce, and the majority cases there the biological mode of the action was not the well characterized. Some of the human proteins that have been the produced with the recombinant DNA technology for the treating of the various disorders of the man. Insulin, diabetes, used for the treatment of the diabetes. Somatostatin, the treatment of the growth disorders. Somatotropin, the growth the treatment of the growth disorders. Factor 8, they can treatment of the use for the treatment of the hemophilia. Factor 9, Christmas disease, is the treatment of the Christmas disease. Interferon alpha, leukemia and the other cancers. Interferon of B, cancers and AIDS. Interferon of lambda, cancer and rheumatoid arthritis. Interleukins, cancer and the immunodiseases. Okay, some of the tumor necrosis factor also they can treatment of the cancers. Abdominal growth factors, these are the substrate and the protein used for the treatment of the ulcers. Next one, erythroprotein is the treatment of the anemia. Okay, alpha antitrypsin, the treatment of the insomnia. Relaxin is the eighth child of the birth. Dexoribonuclease, cystic fibrosis, and is the production of the recombinant vaccines that traditionally either inactivated attendant in the functions of the agents that are the injected in the antibody producing organism to produce the immunity. And again, disease diagnosis, the various techniques of the recombinant DNA technologies can be effectively used in the diagnosis of both the infection diseases and the genetic disorders, employing the gene cloning methods to probe for the diagnosis for all the major pathogens have been developed. Probe-based probe-based modules for the diagnosis of the major pathogens have been developed. Probe-based molecules, acids, molecular acid methods. These are not only rapid but also the sensitive of the specific throughout the recombinant DNA technology. The technology has the facilitated the production of the enough material for the gene sequencing, which is the next one is the gene therapy and the cancer treatment. More than 200 genes are the responsible for the variety of the genetic disorders human beings were identified. At the molecular level, the cause these are the disorders from the specific mutations in this. The genes have also been the illustrated gene therapy aims and the correcting of the defective gene, replacing the defective gene the normal health. Attempts of also being the made of the is the cloning disrupt the infection cycles of human pathogens such as the AIDS viruses. A number of strategies are the being attempted to treat the cancer through the application of the recombinant DNA technology, introduction of the gene for the and antisense RNA copying of forensic science. The application of the recombinant technology in the forensic science is largely depends on the ability of the DNA analysis identify. And next one, some of the popular media, this the techniques are so collectively called genetic fingerprinting. This technique developed by the like Jeffrey is the colleagues is the completely revolution is the forensic biology. The technique of the DNA fingerprinting reveals the great polymorphism, that is the possibility of the two persons having the same pattern DNA fingerprints. This technique allows the identification of the criminals in the murder case, rapist. In the rape case is the murder of the father and mother. Some of the doubtful parentages also they can use for the this technique. Forensic scientists occasionally do the deal with the bodies that the badly disfigured, that the DNA is only for the determining the sex. These techniques will also the identification of sex and the an unborn child. Industrial application, generally engineering microorganisms. Some of the microbial eukaryotes like the Saccharomyces cerevisiae and the Pichia pastries are the now uses the routine production of the several animal proteins. Filaments of the fungi, Aspergillus nidulans and the Trichoderma rixia is also is the expression of the vectors. The genetically engineering animal cell of the animals, genetically engineered the plants, environmental application, biodegradation of the xenobiotics and toxic waste also can use for the recombinant DNA technology. For example, some of the production of vaccines by the transgenic plants, recombinant HBS AAG means that derived from the plant. What are the application? Hepatitis B. Okay. Next one is the cholera, E. coli, heat liability, enteroxin. The main is the plant tobacco, potato, cholera, and the E. coli diarrhea. HIV epitope GP is the main derived from the tobacco and the bean. Application of the HIV viruses. Malaria, malarial beta cells, hepatite, tobacco, malaria, bacterium. The removal of the spoiled oil is also recent years the genetically engineering utilizing of the microorganisms have been the successfully produced, which is the would be grow the rapidly. Okay. Next one, removal of the spoil, detection of the polymer hydrogen, and next one, sequencing of the heavy metals and transgenic plants the poly phytoremediation. Transgenic plants have also been the developed for the detoxification of the soils contaminated with the high metals like the mercury, aluminum, AI, next explosive like the trinitrotoluene. These are transient plants the grown in the contaminated soils. The detoxifying is the contaminants. These are the application of the genetic engineering.